Hello everybody, welcome back to the Computational Genomics Lab. We just finished doing Iranges, where is a function that can keep track of start and stop positions. It was just kind of a baby training example of where we're going to go with genomic ranges today, which is often referred to as GRanges, and we'll see that in a sec. So today we are going to masterclass lessons 08 and GRanges for genomic ranges. And really the main goal here is just to add a little twist onto Iranges where we keep track of the chromosome. So in Iranges we compared two peak files and we saw that 2 of the 3 peaks overlapped. But what if those coordinates, we didn't know we had 22 chromosomes and the X and Y chromosomes. Those start and stops could be the same numbers but on different chromosomes. So if the peak was from 0 to 30, well, that could be on any chromosome. So we just have to keep track of the chromosome and some other features like what genome version are we using. That's sort of built into genomic ranges just to make sure you use the best practices. So a lot of this seems like esoteric kind of name tracking, but it's actually very, very important. So, okay, so let's go to our 0804 RMD file and make our first genomic range. So here we can see that we're going to make the first new thing we're going to see is seq names, and that's the chromosome basically. And so we'll just make one that says seq names is equal to paste chromosome X, and then the strand is either equal to a plus, a minus, or star. So the plus strand would go this way, minus strand would go this way, and a star means the data, I don't know what strand of the DNA it came from. In ATACseq, that's the case. We don't know the strand because the peak, the way the enzyme goes into the accessible regions of the chromatin, you don't know which strand is which. So it's we use the star. But it is important when you look for if these peaks overlap a gene to know if that gene is on the plus strand or minus strand. Okay, but for now we'll just learn that basically you can see we're going to use this irange function again. And these are the same peak values we did in the previous video in 0803. Okay, so that's the Iranges. That's really the same thing we did last time. We're just adding these seq names, strand, and then the ranges is equal to that Irange we did before. So let's give that a run. I see something. Oh, just warning message. Okay. So I can look at this GR, and it's in that sort of S4 object structure. So let's take a look at it more on a high level. So if we look at seq info, we have an object here with the chromosome X. And these are the things genomic ranges usually wants to know and says they're kind of important. Like what genome version is the DNA circular? Like, so for bacteria, they have circular chromosomes. So that could influence how you're thinking about overlaps. And seq lengths are missing as well. And so we're going to add those right now. So we can say seq lengths of the genomic ranges and fill in that slot that currently says NA. So that's just calling up seq lengths of GR, which is currently empty. It's just saying paste in chromosome X is this long. So seq lengths just basically wants to know how long the chromosome is. Okay. And if we do that, we see now we have seq name, seq lengths is circular. We still don't know. We could fill that in the same thing. Same way. Okay, it would be a different thing like seq levels. There's a few things, and the best way to look at any of these things is just to type in genomic or GRanges and put a question mark in front of it, and then you'll get the manual, and it'll tell you all the things you might need to think about with the GRanges. Okay, so we've now added a seq length, and for seq levels, which is again going to be in here, seq name, seq info for more information. It's also written down in this document. Again, in videos I try and go a little faster just to conceptualize some of this stuff. Okay, so we could have, instead of adding each seq names for the chromosomes, we could do seq levels, and that would fill the chromosome spot for us. So we could just paste in as many chromosomes as we wanted to make. In general, you don't make these, or you can make them. It can be important if you're studying a want to study a small piece of the genome across a large set of data, you can make a genomic range. So you just look at chromosome X from this position to this position, and then there's just one window that you can look across more and more data, rather than trying to do all the things and then just pick out that after. Okay, so like we added seq levels, we're going to add a genome, this genome to this GR object, and we're going to just say it's MM10. And now our GRange is complete. And really we're just doing this just to get what kind of information goes into a GRange. I'm trying to make my thing bigger so I can scroll up, but I guess I'll leave it small here. So we can see we have chromosome X, 1 to 30, run length encoded, and we can tell now that this is from MM10. And if we wanted to look at the seq length, we could see the length of the chromosome X, but we didn't assign that for Y. Okay, so we ran the ATACseq pipeline in 08, our pipeline run. Instead of file pathing all the way through it, I'm just going to have this file path here to a peak file. So instead of building toy GRanges and toy Iranges, we're going to do the real thing now. We're going to load all our peaks from our ATACseq analysis and look at all of them and see what that looks like rather than just a couple. Okay, so we're going to take 2 files from the same, or 2 replicates of the same time point. We're going to do all of them later, but for now, just to say, we did this ATACseq. We had this 0 time point, 30, 60, 90, 120, 150. And for each time point, we had 2 replicates. Let's find the peaks that are the same in both replicates, and we'll make a consensus peak file for that time point. So these are peaks that show up in both replicates, which is handy because then it means they're reproducible. Okay, so let's load in one file, peaks, and here we're going to learn a new function, rtrack_layer import. The reason you need the 2 colons there is because import's a generic function across multiple packages. You want to specify rtrack_layer : and that's common for any package, dplyr : and then select, etc. And so we're going to load 2 replicates of the same time point. So we'll call it peaks 1 and peaks 2, and we're going to go to find overlaps. Okay, so in genomic ranges, there's a function called find overlaps. Super handy, because we're going to see here that the length of peaks 1 is 82,184, and we'd previously only worked with like 3. So this is, now we're thinking big, many thousands of peaks across many different chromosomes. We're really stepping up our analysis here. Okay, so let's say the length of file 1 is there's 82,184 peaks, and peak file 2 there's 67,000. So we had less peaks in our second file. Okay, so I didn't do the overlap. So find overlaps is the genomic ranges function that will find the overlaps between 2 GRanges. Okay, so let's do that. There's some warnings there that some of these don't have canonical chromosome names here. Believe it or not, the human genome isn't completely finished, and there's some sequences that have these weird numbers, but we're just going to ignore them for now. Okay, so we did that. Now if we look at the length of OVF, it's 54,000, and that sort of makes sense here. If we look at summary, OVF has 54,000 overlaps. Our sample 1 had 82,000 and 67,000. So 57,000 makes sense that you wouldn't expect a number higher than the smallest number in the comparator. So the smallest number comparator was 67,000, and you wouldn't expect all those peaks to overlap with the 80,000. But what we saw there basically is the 60,000 were all in there, and then there was about 10-ish thousand that didn't overlap between the 2. Okay, and now why does this matter? And I'm not going to go into full detail and consider this an exercise for this video, because the feedback I've gotten is it's super important, but you should really dig into it when it's necessary. And if you really want to know more about all of this, then this is something to dig into a little bit later. But we got lucky that our overlap was less than the smallest number. So let's say we had 3 peaks in one file and 1 peak in one file. How could we get 2 overlaps? It's possible. So you could have 3 peaks and 1 peak. So you'd expect the most that could overlap is that 1 peak. But actually, that 1 peak could be 2 halves of another peak and then have 2 overlaps. Okay, so it's really important to think about where your at_to and at_from are. At_to is the nomenclature for indexing the query file, so this set of peaks, and then the subject file, the other set of peaks, and finds which ones match and overlap. But just to go briefly through this, I don't want to go into all the detail. If we start to look at this here, I'm giving you an example of where I learned how important this is myself. I'm loading these 2 files called crazy peaks, again 2 replicates, and we're going to find the overlap between 2 replicates, but this time we're going to have more peaks than the minimal file, like the 1 went to 2, and how could it be 2 if it was only 1 peak? Okay, so we're going to do find overlaps between these crazy peaks. The length of file 1 was 10,000, the length of file 2 was 19,000, so most we'd probably expect to overlap is 10,255. 11,042. That's a lot more than 10,000. Okay, so how can that happen? And I don't want to go through all the fine details, but this is sort of a pictorial example of what's going on here between at_from and at_to. So if at_to had 3 small peaks in the at_from, it would say I had 3 overlaps. And this one doesn't overlap, and these don't overlap. So here I'm going to say my at_from peak 1 has 3. Peak 1 has 1 overlap, or 3 overlaps. Sorry. So yeah, this stuff gets confusing. So and it's all written out here and spelled out very detailed, because this was the exercise where I learned how important it is. And again, it is a lot of detail, but if you really want to keep moving forward with your computer science, this is 2 solutions to figure out how to think about this. And just as a conceptual solution, what can happen, like we saw, this file had 3 peaks that happened to be, let's say my first replicate had a really big peak, and my second replicate had 3 small peaks called. Well, those are kind of in the same thing, so that counts as 3 overlaps. So that's how you can get a bigger number than the lowest number of overlaps. And so it's good to check the lengths of your peaks, and I go through all the stuff it takes. And it turns out you just start summing these up, so that if I had 1 with 3 and then this with that, that's how you can add them all up and get a number of 11,042. It shows you exactly how to get to that. But I know everybody doesn't want to know all that detail, so we'll just leave it with what we did is we used rtrack layer import to import a peak file, 2 peak files, and we found overlaps between those 2 peak files. And that's a great first step of understanding genomic ranges. And next, we're going to look at genomic ranges of the human genome, features like annotations, where are genes, and then you might have a start and stop of a gene, but within there you might have a start and stop of exons, and you might have a start and stop. So we're going to see why GRanges is really powerful to keep track of what chromosome a gene is on, and all the features associated with the human genome in the next video. All right, hope to see you there.